Terrific.
Hello, everyone. We'll get started in just 1 minute. All right, well, good afternoon, everyone. I'm Diane Hoffman, and I'm on the faculty at the law school here at University of Maryland School of Law, where I direct the Law and Healthcare program. And it's great to see some familiar faces in the audience. Thank you so much for coming this afternoon. It's my pleasure to welcome all of you for the first of our Rothenberg Healthcare Law and Policy series for the 2021-2022 academic year. The series was established with funding from the Karen Rothenberg and Jeffrey Seltzer Law and Healthcare Program Endowment, and it focuses on contemporary issues in healthcare law and policy. As you know, our first speaker is Dr. Eric Green, who I will introduce momentarily, but we also have 3 other speakers lined up for this year. And next, I just wanted to give you a sense of who's in the queue. So next in the series is Professor Lisa Ikemoto, who will be speaking on October 27th. Lisa is a professor at UC Davis School of Law. Her research includes reproductive rights and justice, healthcare disparities, and science, technology, and law. And she'll be speaking, she has a very provocative title, which is The Reproduction of Genetic Race. And then next semester, we've lined up Dean Dana Matthew from GW Law School, who will be speaking about a new book that she's coming out with. And then in March, we'll hear from Ruth Faden, the founder and former director of the Berman Bioethics Institute at Johns Hopkins. And she will be speaking at the end of March. So I hope you'll return for those talks. But let me turn to our speaker for today, who you've all come to hear, and that is Dr. Eric Green, who's has both an MD and a PhD. He is the director of the National Human Genome Research Institute, NHGRI, at the National Institutes of Health. He is the 3rd director of NHGRI and was appointed by Dr. Francis Collins in 2009. But before he became the director, he already had a kind of a long-term relationship with NIH and the Institute. He served as the NHGRI's scientific director from 2002 to 2009, the chief of the NHGRI Genome Technology Branch, and also as in the NIH director of the Intramural Sequencing Center. And prior to that, he also was very involved in the Human Genome Project. Throughout his career, Dr. Green has authored and co-authored over 375 scientific publications. And I could go on and on about his accomplishments, but I wanted to also share a bit about his personal background, I think, which raises the question about nature versus nurture, or maybe makes it obvious about how they combine. And that he comes to us from a scientific family. His father was a virologist at St. Louis University School of Medicine, where he directed the Institute for Molecular Virology for over 5 decades. And his brother, Michael Green, is also an MD, Ph.D., and is a molecular biologist at the University of Massachusetts at Worcester, where he serves as a chair of the Department of Molecular Cell and Cancer Biology. And finally, I should say something about why we asked Dr. Green to speak as part of this lecture series. He has had a long relationship, you might say, with our law and healthcare program through his work with the founder of the program and its first director, Karen Rothenberg, who's our professor and former dean of the law school, who she worked directly with Dr. Green after stepping down as dean of the law school as a special advisor. So it's especially meaningful that he has agreed to give this lecture today. Before I turn the Zoom platform, though, over to him, let me say that there will be some time for questions at the end of his presentation. And if you think of something during his presentation, you can send it to me via the chat function. But if you prefer to ask it in person at the end, you can just use the Zoom hand raising function on your screen. So with that, I will turn it over to Dr. Green.
Well, thank you very much. Diane, can you see my screen now?
I can.
Yeah, great. Well, thank you very much for that kind introduction. And I am delighted to be here today to give a talk as part of this series that is very much a tribute to Karen Rothenberg, a good friend and colleague for many years. I'm going to really just try to give you a very high-level survey view of what it's like at the forefront of human genomics. And you're going to hear about some incredible opportunities that I will just touch on, but I also want to sort of emphasize some of the important societal considerations that really I think are of great interest to this group, certainly for many years have been great interest to Karen, and certainly of great interest to the University of Maryland program that Karen has been such a key architect of for so long. As you heard in the introduction, Karen has been really a very valued contributor to NIH for a number of years, both at the whole NIH level and various parts of it, but also the National Human Genome Research Institute. And in fact, when we were asked to chronicle all of her contributions, she came shortly after I became director, and you heard, and she was my special advisor and really helped me architecturally think through some issues around our bioethics program at the Institute. But that was one piece of a set of contributions she's made. And we had a fun time putting this together in this special issue of the Journal of Healthcare Law and Policy, where we talked about this secret and not so secret set of contributions she's made. And we had to get 4 authors to sort of chronicle it because each of us had had major interactions with Karen. And notice one of the authors was in the news yesterday, Francis Collins, who Karen had significant interactions with over the years, especially when he was director of NHGRI. And of course, he was in the news yesterday because yesterday was the day he announced he'll be stepping down as NIH director. So I'd point you to this article if you want to hear why I am thrilled to be here in paying back some of what Karen gave to us. And I'm happy to pay it forward now by participating in this seminar series. I would also point out, if you want to hear it in Karen's words, some of her interactions with Genomics and some of her interaction with the Institute, we did an oral history. We have a history program at the Institute that I established, and we do lots of oral histories. And one of the earliest that we did was, which is featured on YouTube, was one of Karen, and I was listening to parts of it over the weekend as I prepared for this. And I would strongly urge you, if you want to hear it through Karen's voice, some of her experiences in the field, I would strongly urge you to take a look at that. You know, Karen was a bundle of energy to have at the Institute and to be my partner in thinking through some stuff. And among many things she did in energizing the Institute, energizing me, was to pull her into some of her many passions, but one of which relates to the use of drama as a vehicle for teaching people genomics. And I see Lynn Bush. Lynn Bush is in the Zoom room, and hello, Lynn, good to see you. And she was also a key part of Karen's partner. And I just show you one representative example of multiple times Karen has asked me to perform. And this was at the American Society of Human Genetics meeting. I don't even want to tell you this panel of people she's successfully recruited to basically give a play related to DNA and drama and society. this was a set of all-star geneticists that she was able to recruit in. And I would also tell you that we actually really enjoyed having Karen at NIH helping us think about this as a vehicle as part of improving genomic literacy more broadly. And we even did a workshop, and here's a photo I pulled from that workshop that we had in 2014 on drama of DNA, specifically as a topical workshop. So bottom line is, this has been wonderful to have Karen touch so many parts of NIH, but in particularly the Genome Institute, which I've now been a part of for over 27 years. So what I want to do for the next 30, 35 minutes or so is really at a very high level, just cover three things that I think will stimulate discussion and stimulate your thinking. And I fully appreciate I'm dealing with a very heterogeneous audience with a lot of different backgrounds in science. So I'm not going to go too deep into anything, but happy to answer any questions that you might have. I want to remind you about this remarkable first 3+ decades of genomics, which really should lead us to a very celebratory feeling. But I also then want to tell you about some of the real tough realities we're facing right now. What are the big challenges that we have in front of us, and what are the opportunities associated with it? And then I want to tie it all together by telling you about a new strategic vision that the Institute recently published that I think encapsulates all these things and starts to put a fine point on some of the big societal challenges that we face in the field. So let me first tell you a little bit about celebratory aspects, because I am a proud genomicist. I got involved in the field of the genomics from the very beginning of the field. In fact, the field really didn't even get named. In fact, the word genomics didn't even exist until 1987. That was the year the word was coined and put into the scientific and medical literature in a lead editorial in a brand new journal that carried the name Genomics that described this new discipline. So it's a very young discipline. People sometimes lose track of that. It grew out of genetics, which has some advantages and disadvantages, but genomics is a young discipline. And the reason the discipline was named, the reason the discipline came into being in the late 1980s was this notion that it was time to roll up our sleeves and do something big and crazy and audacious, because we technically were ready to do it. And that was to actually comprehensively study the human genome. That was organized in this big effort that is now in the history books called the Human Genome Project that started in 1990. I got involved as a young trainee, a freshly minted MD Ph.D. In my clinical training and research training phase of my career, got involved on day one of the Genome Project and made a career out of it. Project by any measure was remarkably successful. It delivered the very first complete sequence of the human genome before its 15-year original timetable, did it in 13 years, and did all the other things that were proposed as part of the Genome Project. Iconically, many of you might recall, if you follow sort of the iconic journal covers, these were when the first draft sequence of the human genome was published, now 20 years ago, these were the iconic journal covers that commemorated the public Human Genome Project's effort in Nature. And in Science was published the paper from Celera Genomics, which was the private effort to sequence the first human genome sequence. Remarkably, that was 20 years ago. It feels like it was just yesterday. But this year, both of those journals, in fact, celebrated and recognized that we've now had 20 years of experience of having in hand at least a draft version of the human genome sequence. Believe me, the first 3+ decades of genomics did not stop at generating that first sequence of the human genome. That was just really the first of a set of highlights associated with genomics. And we should celebrate these things. And since this is an audience, I think that's more interested in some of the societal and legal and health policy issues, I'm not going to take you deep into the science of what's happened over the last 20 years, but let me just summarize it in very general terms. Especially from the point of view of the National Institutes of Health and my institute, the National Human Genome Research Institute at the National Institutes of Health, our focus, once the Genome Project ended, related to applications of genomics to human health and to medicine. Now, this wasn't, necessarily sort of, an incredibly creative idea. This was the obvious thing that was going to happen. If you look back when the Genome Project was reaching its end, if you looked in the popular press or you looked in the scientific press, everybody was juxtaposing the two words, genomics and medicine. And the phrase genomic medicine was born. And the idea was, could we get to the point of using the tools of genomics where we can look specifically at an individual person's blueprint, their genome, in a fashion that would allow us to tailor their medical care, to no longer treat people based on average response, but rather to treat them based on specific responses or predictions based on their unique genomic information. So the concept of applying genomics to the practice of medicine was obvious. However, 20 years ago, that concept was there but blurry. We did not really know what genomic medicine was going to look like, but we knew when the Genome Project ended now 18 years ago, that was the aspirational goal with respect to health applications of genomics. This was a journey. The Genome Project was a journey, but the journey to genomic medicine is sort of a second journey. That journey began with the completion of the Human Genome Project, and the finish line of that is when we completely changed the practice of medicine sometime in the future by using genomics routinely as part of clinical care. Now, like the Genome Project, this won't happen overnight. It doesn't happen with one step. It involves multiple steps. We don't know what all those steps are, but we recognize it as a compelling set of things that must be done. And like the Human Genome Project, this is not something that one person's going to do and get a Nobel Prize for. It's not that one country or one funder is going to be responsible. It's not even one discipline that's going to be responsible. This is going, and it's not going to happen quickly. In fact, it's going to take much longer to realize genomic medicine than it was to sequence that first human genome. And so it's not a sprint, and it's not a few participants. It is much more akin to a marathon. Many, many people, many, many disciplines, many, many parts of the world involved, running shoulder to shoulder for a long time. Well, we've been on this second journey now for 18 years. What are the other highlights? I told you the first highlight was getting that first human genome sequence. Well, there have been 5 other highlights, and I just want to just rattle them off to you. And each of these could be a talk in and of themselves, and they represent triumphs of accomplishments that genomicists have brought forth in the last 18 years. Probably the second most significant thing in genomics since the sequencing of that first human genome was that we reduced the cost of sequencing a genome by over 1 million-fold. That first human genome sequence, $1 billion. Nowadays, you want to get your genome sequenced, less than $1,000. Basically the cost of an MRI. Actually, it's probably cheaper than an MRI these days. Sequencing has just plummeted in terms of costs, and that has allowed many, many people to get their genome sequenced and as part of research studies and increasingly clinical care. You give a bunch of energetic genomicists an inexpensive ability to sequence genomes, and guess what happens? We sequence a lot of genomes. We've gone from having one human genome sequenced 17, 18 years ago to now having hundreds of thousands. It's probably already north of 1 million human genome sequences have been sequenced. And what that has allowed us to do is to catalog the 1 out of 1,000 bases that all of us differ. So each of us is 99.9% identical to any other person, but it's that 0.1% that has a lot of important information in it. Most of the variants in our genome, the different spellings, are common in the population, and we now have gone over the last 18 years to have cataloged the most common variants across most of the world's populations, but not all the world's populations. That's another story we'll get to later. And so we have gone from being quite naive about how our genomes differ to having a much better assessment of how they differ. We want to use that information to then know how does that change how our genomes work. Just like you want to know how a typographical change in a sentence may change the meaning of a sentence, we want to know how differences in our genomes change how our genomes function and how that might influence our health and disease. And that in order to do that, you have to know how the genome works. And we've gone a long way in 18 years, got a long way still to go, but we pretty much have gotten a pretty good inventory of the easy parts of our genome. Those are the genes, the parts of our genomes that directly code for proteins, the bricks and mortar of our cells and tissues. But it turns out there's a lot more really complicated choreography and how those genes get utilized during our lives from early development until we die. And there's a lot of changes that go on in how we use those genes. And we've got to figure out all that circuitry, all that choreography, and we're making good progress. But the progress we've made with respect to using these new technologies for sequencing DNA, being able to inventory people's genomic variants, and being able to learn more and more how those variants may influence how our genomes work, have allowed us to really begin to tease out the genomic basis of human disease. Now, some diseases are really simple and they're rare because they're simple because it's one gene that has a typo in it that makes the gene break and therefore cause the disease. Diseases like sickle cell anemia and cystic fibrosis come to mind. And we've done wonderful there. We've gone from when the Genome Project began, 61 diseases that were rare that we knew what would cause those diseases, which gene was mutated. 61 in 1990. Now that figure is almost 6,000. So we have greatly accelerated our ability to understand rare diseases. But most diseases that fill hospitals and clinics around the world are not rare or not caused by one gene. They're caused by multiple different spelling differences in multiple genes, and they're usually more greatly influenced by the physical and social environment. And these are diseases like hypertension and diabetes and all the things that run in your families. And we're making progress, and there's great optimism. Things will get terrific over the next decade, but we still have a long way to go. That's really complicated. But I would say overall, our understanding of the genomic basis of disease has greatly accelerated in 18 years and is setting up a circumstance for the next decade or two that should be breathtaking. And the final highlight of the first 3 decades of genomics is that genomic medicine is no longer an abstract idea. We are beginning to see genomic medicine become real. And that is profoundly exciting. We are gone from having a very blurry idea to actually knowing what genomic medicine might start to look like, at least the tip of the iceberg, even though there'll be a lot more developments. In areas like cancer, we're seeing genomics being used in the clinic. In areas like rare disease diagnostics, we're seeing it used. In pharmacogenomics, picking the right medicine for the person based on their genomic information, we're seeing that more and more. And certainly where we're really seeing it is in prenatal genomic testing, where millions and millions of women who are pregnant are now getting a non-invasive test that involves a simple blood draw to look at the chromosome health of their unborn child. And this is literally done millions of times worldwide now using these new technologies. So those are all very, and we have every expectation that that list will grow of seeing genomic medicine being realized. We are just at the earliest stages of it, but we have a lot to celebrate. So that's my celebratory part of my talk. It is an exciting reality to see genomics where once upon a time, we didn't even know if we'd be able to sequence a human genome. If I go back, a few decades, to now seeing it actually enter medical practice in the earliest examples. But I also want to point out that while we have moved into this sort of DNA revolution or whatever, genomic revolution, whatever you want to call it, and there's a lot of attention to this, that occasionally people might get the wrong impression listening to me that this is all easy and that we don't face any challenges and that my optimistic view about all this is not based on reality. And I want to jolt us back to reality by giving you a few examples of things that we really are tackling and struggling with and are getting a lot of attention. And it really does relate to the theme of this series and many of Karen's passions that have been very much pursued throughout her professional career. So I'm just going to tell you about 3 realities and opportunities, and these are big buckets of things, but I think it'll stimulate some discussion. First reality I do want to tell you about is I don't want to leave you with an impression that just because it is now trivial, and it really is trivial, to sequence somebody's genome, and we really can do this in like a day for less than $1,000, and we don't, we could just do this with a little bit of your spit. We don't even need to draw blood. It is a trivial, and we can even run it through a computer program and give us a report of all the variants that you have relative to some reference. However, I don't want to lead you to believe that if we did that on a patient today and we went unrounded on that patient tomorrow, that we'd be able to look at that list of variants and really know what most of them mean. So we have the technical capability of generating that genomic information on patients efficiently, effectively, rapidly, and yet it is not trivial to clinically understand it. We have a knowledge gap that we're working on. And I just want to really stress that is really holding back a lot of things that we're going to talk about subsequently, because just because you can do something technically doesn't mean that the medical system and the healthcare system, which as you all know is incredibly complicated, is ready to embrace this. And in part because we're not quite there yet. And there's a lot of hurdles we need to get to the point of being able to routinely use genomic information comprehensively as part of medical practice. So that's just a jolt of reality, and I want you to appreciate we're working very hard on this. Multiple major programs around the world to close this gap, but this is one of the grand challenges for this decade. A second reality, and I really feel strongly about this, and it plays directly to this series, is the reality of how much genomics has changed in terms of its relevance. And I've lived through this, and so that's why I feel like it is so real. You know, I remember vividly being a postdoctoral fellow working on the Human Genome Project, and the only people genomics was relevant to were geeky scientists like me who were at the bench, at the computer, trying to figure out how to map and sequence the human genome. It was just biomedical researchers, and nobody else really was particularly relevant because it really just didn't permeate out. I think 18 years ago, when the Genome Project ended and we said, let's put genomics and medicine together, our tent expanded considerably. We were successful in bringing healthcare professionals to come in and join us as part of a research enterprise to try to imagine how genomics might eventually be applied to medicine. And with that came a lot more complexity because now we were dealing with healthcare implementation, which is incredibly complicated. But I think what's really changed over the last 5 years relates to the last highlight that I gave you a few slides ago. And that's the fact that genomics has now touched medicine and healthcare. And with whether it's cancer genomics, pharmacogenomics, rare disease diagnostics, or prenatal genomic testing, genomics is becoming relevant to patients. And once it becomes relevant to patients, it becomes relevant to their families and their friends. And all of a sudden, that means genomics is relevant to society. And so this comes with great responsibility, great complexity, and an immense amount of opportunity for study, but one that we must be very thoughtful about. Now, I don't want to mislead you to think that we never knew this was coming, that day wasn't coming. My main message is that I think it just came faster than I ever could have anticipated. And the reason I say that we knew it was sort of going to come eventually is we did have a proactive stance in genomics. And dating back to the beginning of the Human Genome Project, and to the credit of the founders of the Human Genome Project, was a recognition that the Genome Project, what it was doing was so fundamentally important that we needed to proactively study the ethical, legal, and social implications of the advances in parallel with making those advances. This, of course, gave rise to this thing called the ELSI research program, Ethical, Legal, Social Implications Research Program, that has now existed since the beginning of the Genome Project and is now part of our authorization as an institute at NIH, where we dedicate 5% of our research budget to this program. And many of you are familiar with it, and some of you are even grantees, I believe. So I look across some of these names. And so this has been a very, I think, successful program in bioethics. It's probably the biggest pot of bioethics research money, I think, in the world. And it's something that the Institute and the field, I think, is very proud of. This, of course, underscores Karen's interactions with our institute and touches on many of her professional passions for many years. The obvious one that comes immediately to mind, of course, is genomics and the law. And of course, I'm just going to mention these topics. Every one of these is a major academic field in and of itself. But we've seen a lot happen in this area, as many of you know, related to the patenting of genes and Supreme Court rulings, et cetera, et cetera. But there's many other things where genomics and law interface represents productive areas of research, great societal challenges, and opportunities for improving how genomics is going to be used in society. As I mentioned, the delivery of healthcare, it is incredibly complicated. It's incredibly broken. That's true in America. It's true in every other country. It has been very interesting to watch genomics be implemented in healthcare from an international perspective. And of course, I have many genomics colleagues from around the world, and every one of them have a different healthcare system, and every one of them complain in a different way about why it's so hard to implement and change healthcare. And of course, it is hard. We are very much in the crosshairs of developing the evidence base in the US to get things paid for, to get things properly adjudicated, to get it as part of practice guidelines. Many, many complexities here. Our approach, of course, is to direct our ELSI research efforts to help knock down the barriers that are in the way of healthcare delivery when it comes to genomic medicine implementation. Among the many things to consider, of course, is regulatory oversight. This genomics, like any other part of healthcare, has to have regulatory oversight. This is a topic in and of itself. Books are written about this, and many, many, many studies and efforts are underway to smooth the path to see genomics become a reality in medicine. There's, of course, also concerns on the patient side of things, many concerns related to many things. Among them, of course, is privacy. And there's no question that, like many other aspects of our life and many other aspects of our health information, it's hard to imagine anything more intimate than our genomes and the genomic information. And so many, many challenges, many issues, and it particularly becomes tricky because the incredibly important aspect of genomics that sets it apart and has accelerated its progress has been data sharing, has been sort of a change of culture where data is shared on a large scale well in advance of when it traditionally was shared. When you start to get to human genome sequence and you're large scale sharing data, it starts to raise issues around privacy and concerns of the research participants. This gets very complicated very quick, but we can't ignore it because we need our research participants to be partners with us on this journey, and we need to strike that right balance of protecting them, but also sharing the data. Part of it, of course, is making people realize the importance of genomics as part of their everyday life and as part of their health care. And to be able to get them to appreciate, we need to be cognizant of the literacy of genomics. It can get complicated, and any health issue is complicated for many patients, and genomics is no exception to that. So this is something we think a lot about at the Institute and have major programs to try to facilitate advancing genomic literacy across the board, patients and professionals alike. And of course, I'd be remiss, especially through the lens of today's society, to not appreciate the fact that there's many issues around health equity and many issues around disparities, and too many examples of health advances only benefiting some. And if genomics ends up like the same as these others, I will be profoundly disappointed. So a significant amount of attention within the genomic society space to think about health equity and the reduction of health disparities, or at least not exacerbating health disparities. So it is on that note that I think if I was going to highlight a new reality that is jolting but important to appreciate, I just wanted to sort of expand on one of those things I just gave you, because I do think this is an area that needs significant work. And so the third reality and opportunity I want to tell you about is not one that I'm particularly proud of, but the fact of the matter is there is a diversity problem in genomics. Now, there are diversity problems in many parts of the society, but I will tell you, in genomics, we are not doing well, and we must do better. To put a fine point on that, I would point out this is actually well written about. Even back 5 years ago, this was started, and even before then, it was written about that the individuals who were getting studied in large genomic research projects were disproportionately individuals of European ancestry. And there was sort of an alarm that went off in about 2009. And in 2016, when this article, you can see that the blue part of the pie chart, it did get better. It got bigger, a little less European from 2009 to 2016. The unfortunate part is that most of that expanded blue part represented China getting into genomics on a very large scale. So it was almost all that blue was Asian, which was great, but it still wasn't as diverse as the world population. And so to really put a finer point on it on more recent data, this just represents all the people that have participated in large-scale genomic studies. And in red, either in numbers or on top or as a percentage below, you can see that, Europeans are overwhelmingly overrepresented, because on the far right, you can see what's the world distribution population of these different groups. And you can see that genomics is still heavily European ancestry. That is a significant problem. And it's a significant problem for lots of social reasons. It's actually a significant problem for a lot of scientific reasons. And this is something we are working very hard to correct. There's one other subtle point I want to make, because I do think it's important to consider. Everything you're looking at on this slide was scientists basically doing this to ourselves. I mean, we were simply not recruiting enough diverse participants. So we ended up with a complete skewing of the representation in our data. Here's where things risk getting worse, is that the projections are that over the next 5 years, we will be victims of our own success, in that because we're seeing genomic medicine being implemented, we will likely see far more genomic data generated by healthcare than generated by research. And healthcare, the scientists can't control. That's whoever goes and gets their genomes analyzed as part of routine healthcare. Well, who's the most likely to get cutting-edge genomic-based medical care? It's most likely going to be those who have the means to do so, and those are likely going to be disproportionately those from European ancestry. And so all of these things might get worse, because our successes in genomic medicine might see more data generated as part of medical care, which might actually exacerbate the diversity problem. So this is something we are acutely trying to get ahead of. We have some catch up to do, but I just wanted to point out to you the kind of societal issue that we are currently grappling with and how we're really trying to fix this problem, but it does not happen overnight. So with that as sort of a celebratory first part, sort of a little bit more of a sobering second part, I thought I would end my talk by just telling you some things about our new strategic vision. And here I want to remind you, and I think the middle part of my talk really did bring this to the forefront. And that is that, you know, I may show some very pretty slides that make it seem like these incredible achievements happened easily, but trust me, the journey of the Human Genome Project, the journey to seeing genomic medicine become a reality was not a linear path. It was lots of twists and turns, and it always requires reassessing where you are and figuring out the best path forward. In fact, there's sort of a culture in genomics of bringing along new ideas every so often, and in fact, the history of genomics, starting with the Genome Project, and then as genomics sort of diversified out across all of biomedicine, really had associated with them landmark publications, 3 of them that guided the Human Genome Project. And then when the Genome Project ended, NHGRI has basically put out 2 strategic visions that have guided the field of, at least for human genomics, over the last 17 years. And so these sort of strategic visions help sort of recalibrate and then recast the vision for what needs to be done for the subsequent years. And so as we sort of got near the end of last decade, by about 2018, we were looking at our strategic plan that had been published 7 years earlier and said, you know what, it is time to come up with a new strategic vision. And it's particularly time because sort of the whole field of genomics had changed. Sort of the threads of the Human Genome Project and the success of sequencing that whole first human genome, sort of over the last 17 years have gotten woven into the fabric of biomedical research and medicine and even society. And it's getting more and more clinically oriented and more complicated. And in fact, as we thought about how we needed to have a new strategic vision, we also realized that genomics was much bigger than just our institute, and we needed to have a vision not just for all of genomics, that was too big for us. And we even rebranded our institute and now really focus on the forefront of genomics. And so it was sort of looking at the forefront of genomics that we set forth from 2018 until 2020 to come up with a new strategic vision. And we went through and did all sorts of things, about 50 events to engage the community. And I'm not going to tell you much about those details except to point you to the final product of it. If you're interested in the details, go to this website shown on the right side of the slide. You can also get the strategic vision. But at the end of the day, we consulted with thousands of people and over 50 different events and lots of other ways of giving us input. And we published in 10 pages of Nature a strategic vision for improving human health. And as we sort of guided it at this mantra of the forefront of genomics. So I'd really encourage you to read these 10 pages, but let me just briefly tell you what the major components are, and then I want to show you a short video, and then quickly tell you about one other thing. But what was very interesting about this round of strategic planning was that what we heard from the community was very heterogeneous. It wasn't all about doing big research projects. Some of the things we were told related to how we needed to lead at the forefront of genomics by being responsible stewards of guiding principles and values, some of which I went over earlier, that sort of undergird the field. We heard from the community that being at the forefront of genomics was being a responsible steward of the foundation of genomics that everybody relies on. We needed to sustain that foundation. We needed to build on that foundation. We heard that the forefront of genomics involved being responsible stewards to knock down the barriers that are hindering progress in genomics. And we heard about examples in the past, like knocking down the cost of sequencing a human genome 1 million-fold. And they said, we need other barriers to be knocked down in a similar way. And part of our plan describes those as well. And we also heard about audacious research projects, as always, that needed to sort of deal with some of the grand challenges. And all these things together represent the forefront of genomics. But the other thing that we had fun doing that I want to share with you, because I think it elicits some conversation during the Q&A, is we decided to end our strategic vision doing something we've never done before in any of these previous strategic visions, where we got together and we came up with 10 audacious, inspirational, aspirational, bold predictions, things that probably will never come true in 10 years, but maybe a couple of them will. And this got a tremendous amount of attention when we published the strategic vision. And in fact, we used it as a vehicle for promoting our new strategic vision. So I want to share with you now a brief video. You'll recognize the narrator. He's a famous author, but we'll go through these very quickly, these 10 bold predictions associated with our new strategic vision.
Genomics, which is the study of all DNA in an organism called the genome, is changing the way we practice medicine, how we manage and treat some previously incurable diseases, and our relationship with society at large. Since the 1950s, double helix and DNA have become household words, standing in for the historic discovery of the 4 chemicals that give us life, commonly abbreviated as A, C, G, and T. Genomics is at an age of innovation and invention like no other, all thanks to how strings of those 4 letters encode the information for creating and operating something as complicated as a human body. Springing forward from the success of the Human Genome Project, a massive reduction in the cost of sequencing a human genome, and the dawn of the CRISPR gene editing era, genomics is leading into an even more audacious future. A future that will define our collective lives for decades to come. But what could this future look like? Here is a summary of 10 bold and fantastical predictions made by experts at the National Human Genome Research Institute and the genomics community for the next decade of human genomics. While most are unlikely to be fully attained by 2030, achieving even one or more of these by that time would have lasting impact for science, medicine, and society. Prediction 1. Sequencing and analyzing a complete human genome will become commonplace for any research laboratory. Prediction 2. The role of every gene in the human genome will be known. Prediction 3. Environmental influences on our genome will be routinely used for making predictions about health and disease. Prediction 4. Genomics will no longer use social constructs such as race in human research studies. Prediction 5. Student projects involving the study of millions of people's genome sequences will be regularly featured at school science fairs. Prediction 6. Genomic testing will become commonplace in medicine, just like a standard blood test is used now. Prediction 7. It will be readily known if a given letter difference in a person's DNA is clinically important. Prediction 8. A person's complete genome sequence will be available on their smartphone in a user-friendly form. Prediction 9. Advances from human genomics will benefit all of society and not just a few. Prediction 10. Genomic discoveries and technologies will help cure more genetic diseases than ever before. How do we get there? The National Human Genome Research Institute is committed to leading research to make such predictions a reality. Learn more about the Institute's 2020 strategic vision for the future of human genomics by visiting genome.gov/2020SV. Dive deeply into a field that continues to transform medicine and will hopefully continue to bring curiosity, purpose, and compassion for people everywhere.
Well, I hope you enjoyed that video. I will also tell you that we got so excited about these 10 bold predictions. We actually have a seminar series that I guess we've just gone through 9. No, we just did our 8th one. We have 2 more. And I would send you to, and all of these are being video recorded, where we're taking each of the predictions and having 2 speakers come and talk about them and about their research and about these predictions. And it's been a very fun series. All of them are available on YouTube. If you have any trouble finding them, just send me an email. But we really are getting a lot of miles out of these 10 bold predictions. Well, with a strategic vision, you should recognize that putting a strategic vision together involves collecting lots of input of ideas and needs and proposals and obstacles and synthesizing that and then writing it up in 10 Nature pages. But of course, it's much more comprehensive than we could even immediately execute. But a subset of the things we do select and now put out in the form of new projects and new programs and initiatives and policies. And this will be what we do for the next 10 years at the Institute. And I could be telling you about many of these different things we're doing, but I really wanted to bring this back to one of the ones that we think is amongst the most important. We heard overwhelmingly during the strategic planning process, far more than we've ever heard before, were these great concerns about diversity. Remember number 3, the reality I told you about, and issues around equity. But in particular, we heard loud and clear that this needed to undergird almost everything we do, and we need significant attention. For example, we actually wrote up the strategic vision in the section on the guiding principles and values. We mentioned among them the need for champion a diverse genomics workforce, because the promise of genomics cannot be fully achieved without attracting, developing, and retaining a diverse workforce, which includes individuals from groups that are currently underrepresented in the genomics enterprise. Not surprisingly, by the time we got to the robust foundation for genomics, it appeared again, and we talked about the need that part of the foundation of genomics must be the fostering of a diverse genomics workforce, and we describe it, I'm not going to read it, but we describe it in a little bit more detail. We are putting our money where our mouth is when it comes to this issue. And so when our strategic plan was published in October of 2020, the very first week in January of this year, we released a new action agenda, as we called it, for improving and building a more diverse genomics workforce. It's an action agenda that lays out the components of it. The key architect of this, working with others at the Institute, was Vince Bonham, who's now my acting deputy director and a long-term expert in genomics and health disparities. And Vince and I wrote this commentary, which was the very first paper published in 2021 in the American Journal of Human Genetics. In that perspective and in this action agenda, which you can download from our website, we outline some of the things we are going to be doing. We are now putting out funding announcements along those lines, and we are putting money to try to do what we can, including building partnerships with not-for-profits and with other institutions to try to move the needle on improving the diversity of the genomics workforce. I remain active in this space. A couple of weeks ago, I published an op-ed in The Hill, and you go to that, you'll find it by Google. And I just want to point out that it also goes beyond just the workforce. I talked about in this op-ed how my NIH leadership team and I have been frustrated by the lack of diversity in both the field in general and genomics workforce specifically. In the last year, we have been critically introspective about the changes that we must make. Making genomics more diverse goes beyond just enhancing the diversity of the workforce. Such efforts must also focus on the broader diversity of the research enterprise, including research participants, as I showed you data for earlier. The character of a scientific field affects all it touches, and funding agencies such as NHGRI have a duty to build an anti-racist ethos into everything that we are given, we do, given our influence and our reach. So we are continuing to put our voice behind these ideas, and I think it's something I would imagine be of great interest to many of you. So I will stop there just by pointing out that we think this new strategic vision represents the latest chapter in the series of visions that have guided the field successfully for the last 30 plus years. And we really look forward to executing on many of the things that we describe in that Nature paper. And in closing, I would just invite you to stay connected with me and with the Institute. You can stay connected with me if you're interested in a newsletter that my staff and I put together and publish once a month. If you want one extra email a month, sign up. If you're more on the social media side, I have a very active Twitter account now that I've had for a couple of years. We put out tweets every day, every work day. And a lot of the things we're doing in many of the things I talked about, we communicate through this newsletter and through social media. So with that, I will stop. Thank you for your attention. I will stop sharing my screen, and I will be delighted to answer any questions you have.
Thank you so much, Dr. Green. That was fabulous. So informative and comprehensive. So I don't know if people have questions they want to put in the chat, or if you raise your hand using the Zoom function, you can go ahead and do that. But while people may be thinking about that, I have a number of questions, but I'm just going to pick up on the last things you were talking about diversifying the workforce. And I guess I'm going to ask you, isn't this a broader issue about science education in our population and attracting people into science? And it seems already that that's just such a tremendous goal of a problem with our population. We don't do a good job of science education. And I just kind of wonder how you.
Well, no, you hit the nail on the head. There are, and we also have to appreciate, we're a relatively small institute. We're at a research organization, not an education organization. So there's even limits to what we can do. The good news is all of this is also happening at a time where NIH is very much introspective, an introspective mode of looking at what we are doing wrong in terms of diversity more broadly across biomedical research. So the efforts we are going to focus on are going to be specifically focusing around genetics and genomics, where we have seen other disciplines improve their workforce. And so we think with a focused attention, we could do much better. And this applies, we're going to cast a wide net and try to hit a lot of different levels, but even if you take something as simple as medical geneticists, it is not a good situation with respect to diversity. And that is, we believe, even a modest effort could go a long way of helping to improve that circumstance. And that's true in the genetic counseling space, it's true in the medical geneticist space, but also we fully appreciate that, and it's part of what I talked about in the op-ed, if we don't fix this problem, we're going to not be able to necessarily easily fix the diversity problem. That, if research participants often will say they will feel far more comfortable working with people who they feel, are more like them. And we're not going to fix diversity if we don't start having our workforce look a lot more like the diversity that society presents to us. And so these things are very much interrelated. So we're gonna target this and see what we can do. I see somebody with their hand up.
Yep.
It's very polite. I'm happy to go to Natalie.
So I have a kind of self-interested question. I have been doing research on newborn screening and genomic and genetic analysis as part of newborn screening. And so one of the debates I run into a lot is how realistic is it to think that genetic analysis will actually come to occupy a central role in newborn screening, which up until now hasn't done a lot of direct sequencing. And so, as part of the strategic plan, or the kind of 10 goals, you talked about innovations in preconception genetic sequencing and analysis, prenatal, through NIPT testing, childhood, and other kinds of genetic analysis. And so I'm wondering, what's your view on the future of genetic sequencing in the newborn screening space? I will admit, candidly, I'm quite ambivalent about it.
So it's actually funny because the four of us at the Institute have a piece submitted to a journal that's sort of an editorial kind of a perspective, and we're waiting to hear back from the editor when they're going to publish it. But we speak exactly to this issue. And basically, my view, which is what we, all of us agree, or at least the four of us that wrote this agree, is we don't think it's a matter of if, we only think it's a matter of when. I think we will. I am firmly of the belief that, and in fact, shortly after I became director of the Institute, I started and we completed a program called EnSight, which you probably are familiar with, I suspect, where we looked at this very deliberatively, and the only area where it showed that it absolutely was here and now, and is now, I think we really moved the needle, is in the neonatal intensive care unit space, where if you have an acutely ill neonate and if that child can be predicted to not do well over the next 3 days, they can get a genome sequence in 24 hours if you're at a hospital that can provide that. So that, but more broadly, not yet. However, it is my opinion, and this is what this perspective says, is that as things change and as we find more uses of genomic information for pharmacogenomics or for polygenic risk scores, and I know I'm getting geeky and some people may not totally know what all that is, but basically the more uses you start to have, I think we'll reach a tipping point where economically it will make more sense just do it at the beginning of, at birth, and have that available rather than doing it and be delayed and have it on an ad hoc basis. And just as newborn screening, sort of has now fallen into routine practice, in most parts of the developed world, I think and because it's cheap and because it's well accepted, I think we'll reach that point. I don't think we're there yet, but we can argue about whether we're 5 years away, 10 or 20. I think it's inevitable. And the more uses later in life a genome sequence plays, I think the more it'll just become an economic argument. Just get it done. So, Robert?
Hi. I was interested in your comment about where the data is going to be coming from in the next 5 years, more from clinical settings and less from formal research settings. And then you mentioned that those with access to healthcare will then be represented, which is a perennial problem in this country, not so much in other developed countries. And I'm wondering whether you think we'll start to get more diverse data from other countries that have healthcare systems more open.
One would hope. The most progressive country in this space that I have great admiration for is the UK. The problem is they're not terribly diverse, so they're not going to, and so that's not going to help us there. And there's significant energies being placed on efforts to standardize the data collection and also to share the data. So yes, there will be some efforts where more data, but the hope is that they'll share the data. There's no guarantee of that. And every country handles their data sharing differently. So that's the other challenge we're facing. But that would be wonderful if we can get some of it. But the fact of the matter is, if we're ever going to truly see the blend of our society here in this country, we need to just take responsibility of get the blend of genomic data from that represents exactly what our country represents. So I don't think we can avoid it. Now, again, one thing I cut out of my talk, because just for time, was any discussion of these large cohort studies like the All of Us research program, which many of you are familiar with. And that's a big program at NIH. 1 million people are being enrolled, and they are overwhelmingly enrolling people who are traditionally underrepresented in biomedical research. If that all comes to pass, they may help fix a lot of that problem, at least from a data point of view. But in the long run, in the long run, if we're successful, still most of the genomic data will be generated as part of healthcare.
Right. Do you think there's going to be a difference in willingness to share data?
Well, that's the next question, right? And so there, the big debate is, and here in America, right? If you've seen one healthcare system and one set of rules, you've seen one healthcare system and one set of rules. And there, I think the big discussion we need to have is who controls that data. Of course, my attitude would be it should be the patient. And even the thing in the video and our bold prediction is that my attitude is it's your data. It should, if you want it on your smartphone, it should be on your smartphone. You want to give it to somebody, you should be able to give it to somebody. And whether it's your CBC values or it's your genome sequence, it's your data. And I think we'll have a reckoning about this, about who owns information, who owns medical information, who owns somebody's genome sequence, and then people will vote with their feet. And they'll either share that data for research or they won't. But along the way, we better not lose. First of all, we have to gain the trust of those who we've lost, and we need to retain the trust of those that we have. And neither of those is simple.
Thanks, Rob. I've gotten a couple of questions to me in the chat. We only have a few minutes left, but one of them has to do with research and the development of new drugs. And as we move towards precision medicine, I guess, and fine tune and tailor these drugs to very specific populations, how will that affect kind of what's been the gold standard, the randomized controlled trials and the large need to do kind of large phase 3 trials? Will we, or what's the thinking now, I guess, about that?
Well, I think we're in the midst of raging debates about this, and I think we're going to learn a lot over the next 10 years. It's my opinion that 10 years from now, the idea of doing any clinical study without having genomic information on people will just look old-fashioned. But there's and it is the hope that by doing so, there might be medications, or there might be possible treatments that would otherwise flunk out because it just doesn't look like it's working enough. If you would only restrict it to certain people with certain genomic signatures, it might be able to save that medication, from basically being ruled out. So but that hasn't been proven enough times. It's sort of been proven in cancer to some extent, but that's sort of the hope. is that we will be much more genomically oriented in the way we do science. And some of this came to the fore even for some of the COVID things going on now. That it's very clear that if you have genomic information is much more informative in interpreting some of this the results of COVID trials. Karen has her hand up. She should certainly have the right to ask me.
Yes, I think Karen Sharp probably be able to have the last question or comment. Oh, Karen, you're muted.
First of all, I just wanted to thank you for just such a great presentation. I just wanted to give you a hard time, as I always like to do, with Natalie's question, just to follow up, because it really scares me to think that we could see a time when we're going to just routinely do whole genome sequences on every newborn without all the other things that need to come with that. We can't even hold together the current newborn screening. We can't get, for example, for PKU kids that are poor, we can't get them the diets they need. So even in the circumstance where we can understand what the genome means, how is it not going to make the equity issues worse?
Well, I mean, first thing is, my caveat was that it's not a matter of if, it's a matter of when. So in my optimistic view will be that the when will be when we think we have as many guardrails up as possible, but they'll never be perfect. It's not even perfect now. So,
right.
So you're absolutely right. Even with newborn screening, there's still disparities. I don't think it's fair to expect genomics to have a difference. I think we are going to work so hard not to exacerbate disparities, but I also don't think we should hold back because of some concern that there won't be any disparities.
No, of course.
So I just think we're going to have to balance that. I predict if there's enough examples of pharmacogenomics for very young children that could be clearly part of routine medical care. You'll have enough examples where you'll say it's just not right that we didn't have that information. That poor child would have never had that horrible drug reaction had we have genomic information. And I mean, as you well know, the genetic screening, the newborn screening that's done, it's somewhat arbitrary what's included on that panel. It looks, the fact that most states it's still in the dozens, and yet we know the genomic basis of 5,700 rare diseases, that seems miles apart. It just seems that we will reach a point that it just doesn't seem right not to get the information. But you're absolutely right that if we start doing that, if we don't have the other things in place to help so that not to exacerbate disparities, then we're going to be shooting ourselves in the foot. But
sounds like an LC research project.
It sure does. It sure, but again, I don't think that should be any. There are some differences, but there's mostly similarities with other implementation of other technologies in medicine.
Agree.
And by the way, before we're going to sign off, I just want to say hi to Barbara Fuller, who I haven't seen from a long time. And I see on my, this is the most I've seen Barbara in months and months. There are many other good friends I see in the Zoom room, but she had her camera on the whole time, so I really wanted to reach out and say hello to her. So
me too. Hi, Barbara.
You're only getting younger. What's going on?
Me too.
Well, we've come to the end of our hour, and again, I want to thank you and thank all of you in the audience for coming and listening. And we hope that you will follow Dr. Green and his work at NHGRI, and also come back for our next lecture with Lisa Ikemoto at the end of October. So again, thanks again for coming. Thank you,
everyone. Very nice spending time with you.
Thank you.
All right, bye-bye.
Bye-bye.